So today I will talk about a new promising test for intravital diagnosis of Creutzfeldt-Jakob disease. So we are not new in the field because a long time ago we decided to see whether the olfactory pathway was involved in prion disorder. You see, this is the olfactory epithelium, which is located in the nasal vault, and these neurons gives their first relay in the olfactory bulb, which is the intracranial structures, and then sending a pathway to different brain structure. At the beginning, we were very surprised because all the cases studied at autopsy, we sampled the olfactory structure, including the olfactory bulb and olfactory tract. And you see here, the brown part is the prion deposition, and this is the Western blot analysis. So the point is that invariably we detect the pathological form of prion. So the point is that we say, but if the olfactory bulb is involved, why not the olfactory neuroepithelium, which is located here? So this is, you see here, the histology of these neurons, which are located in the nasal vault. These are a unique tissue in the nervous tissue because these neurons are ongoing to replication. So these basal cells became mature, and they become mature neurons. They send axons to the olfactory bulb, which is intracranial. The first point is that olfactory mucosa had prion. Okay, you see here the black part, it is the prion protein. So this is the first study that was long ago, 2003, and this is the first 9 patients that we studied that we took the olfactory neuroepithelium at autopsy, and you see here the brown part, it is the deposition of the prion protein, pathological form in the nasal vault. And this is the Western blot analysis. And this is here, you see the immunoelectron microscopy. These are the cilia of the neurons, and these are the gold particles which stain the prions, which are in the olfactory neurons. Another important issue is that if we detect patients with different strain of prions, like John Collins explained previously, we found the 2 different prions into the olfactory neuroepithelium. So, spurred by this result, we start to doing biopsy. And, you know, using these forceps, we enter into the nose and we collect little specimens of olfactory tissue. We did the immunocytochemistry, and you see here the positivity in the cilia of the olfactory neurons and in the basal cells, while Alzheimer's disease control were completely negative. The problem is that we have a lot of concern about the olfactory mucosa biopsy. First of all, because it was an invasive procedure, and surgical complication might occur, such bleeding, damage of lining fibrosa, and other complications. The other point is that the low amount of tissue precluded the biochemical analysis, and we did only immunocytochemistry, which sometimes provide inconclusive result. And the other point is that the olfactory mucosa is not completely neurons, but it is interspersed with the respiratory mucosa, which is negative for prion. So we thought about to do a brushing. A brushing is not invasive, and it provides an efficient sampling because we collect simply the outer part of the epithelium. So we didn't have to take the whole epithelium, just the neurons which are located at the surface. So the point was, how can we bypass the immunocytochemistry? And we saw that there was the RT-QUIC that was a very interesting method because it was 100% specific and 80–90% sensitive in the CSF of CJD patients. So we combine the olfactory brushing with RT-QUIC to do this study. So the olfactory brushing is very simple. We put a tampon into the nose. You see, this is a fibroscope which allows to see the inside of the nose, and we get into olfactory and we collect neurons just rolling down this tampon. And if you see here, this is an example of olfactory neurons that we detect by a simple nasal procedure. So we did the study to see whether the olfactory brushing coupled with RT-QUIC might be an in vivo test. So we took the first 31 CJD subjects, and in this subject we collect both CSF and olfactory mucosa to see whether we might change the sensitivity, which was 80% on the CSF, and we didn't know about the olfactory brushing. And we have also 31 normal controls and 12 patients with other neurodegenerative disorder. So this is an example of the first 14 patients. We see here we had all different genotype at codon 129 of the patient. This is the clinical signs, and this is the CSF result, and this is the final diagnosis. In this graph, you may easily understand the RT-QUIC comparison between the olfactory mucosa and the CSF sample of the same patient. You see the red line are the CSF, and the blue line are the olfactory mucosa. As you can see, for example, in patient number 7, we didn't have RT-QUIC seeding activity in the CSF, while it was, really high in the olfactory mucosa. This happens even in patient number 10 and in patient number 12. So you see the two curves, which are completely different about the seeding activity. And all the controls were negative. So this is the result of our study. So we collect the olfactory mucosa on 31 subjects, and 30 out of 31 resulted positive, while the same CSF samples from the patient were 23 positive out of 30. So, here we have a sensitivity of 97%, and here is around 77. So the two tests provide, of course, significant different results. And this is the control that were all negative. You see both in the CSF and in the neurodegenerative disorder. So we did even a preliminary analysis of the genetic study, and I think that Christina will show you later. One was CSF negative, while 2 out of 2 were positive in the olfactory mucosa. Here you see the graph representation of the olfactory mucosa curve and the CSF. Okay, you see that they are really significant difference, both in terms of seeding activity and positivity. And here you see the olfactory mucosa, the intensity of fluorescence, and this is the CSF. And this is the negative case of CSF. You see there are 7 here, while only 1 in the olfactory mucosa. So the conclusion is that the detection of seeding activity in the olfactory brushing provide a 97% of sensitivity and 100% specificity, and indicates by provide a strong basis for definite intravital diagnosis. So the relative high seeding activity in the olfactory mucosa might suggest biosafety implication, but open a new window of study, because other neurodegenerative protein misfolding disease which involve aggregation might have misfolded protein in this structure and might be novel candidate to be studied by olfactory brushing. So I would like to thank our group in Verona, my collaborator in particular, Salvatore Monaco and Matilde Bongianni, who together with Dr. Cowi's laboratory and of course Cristina Roux, we performed most of the study. I want to thank also Giovanni Tonali, who is the otolaryngologist who we did together all the sampling in the patient, and Maurizio Poccheri and the National Institute of Health, which give us an important effort to collect the patient. So we deeply acknowledge also the families for their consent to perform olfactory mucosa, and all the colleagues which help us to pursue this study. So thank you for our attention.